Hi everyone, my name is Marianna Rapsomaniki and I'm excited to present to you today our work on a stochastic hybrid model of DNA replication that incorporates protein mobility dynamics. This project is a result of a collaboration between our group at IBM Research in Zurich, the Automatic Control Lab of ETH Zurich, and the University of Patras in Greece. DNA replication is the process of duplicating the genome before cell division and forms the basis of genetic information maintenance. In eukaryotes, DNA replication initiates from multiple sites along the genome called replication origins. During the early G1 phase of the cell cycle, selected origins are licensed, and during the G1 to S transition, licensed origins are activated by protein particles that trigger origin firing. Firing leads to the formation of two sister replisomes on two replication forks that start unwinding the genome and synthesizing a new copy, and the process continues until precisely two identical copies of the genome are produced. DNA replication is a complex and uncertain process. Origin firing is stochastic, and this concerns not only which origins will fire in each cell cycle, but also when they will fire. Recently, DNA replication timing has been observed to be highly correlated with the three-dimensional structure of the genome and nuclear architecture. But how exactly does chromatin folding affect replication timing? In this work, we set out to investigate the interplay between DNA replication, three-dimensional chromatin structure, and protein mobility dynamics. We developed a stochastic model of DNA replication that exploits 3D genome structures from the literature to simulate the spatiotemporal dynamics underlying origin firing. We tailored the model to the case of fission yeast, whose nucleus you can see here, and our model consists of two interlinked modules: a particle-centric module that models the mobility of firing factors within the nucleus, shown here in different colors, and an origin-centric module that captures origin firing and linear progression of replisomes along the genome. Now, in contrast to existing work, our model explicitly models origin firing as the interaction between firing factors and origin sites. When a particle meets the attraction sphere of a licensed origin, it binds to it and splits into two monomers on two replication forks that move along the DNA. Origins that have not yet fired when they are met by an incoming fork get passively replicated, and when forks meet, a new particle dimer is formed that is released back into the nucleus to diffuse and potentially activate other licensed origins. So what I'm showing you here is simulations of our model where the dots correspond to newly fired origins, the lines of different colors correspond to the three different chromosomes of fission yeast, and as the process goes on and DNA is replicated, you see that the lines grow, they become bigger until they finally merge. And the process continues until approximately 20 minutes, which we know from the literature to be an estimate of the duration of a full genome replication in fission yeast. We simulated the model for different values of the parameters, different structures, and under different hypotheses, and collected a total of 300,000 in silico profiles. We first examined the spatial organization of replication. The model was able to reproduce experimental evidence that I'm showing you here in the first column, only when particles had to diffuse to the SPB to get activated. We then went on to examine this in vivo using immunofluorescence and live cell imaging. And what I'm showing you here are fission yeast cells that have been stained with HEXT, that marks total DNA, MIS6, that marks the centromeres, and IDU, that marks newly replicated DNA. We did this process for both wild-type cells, but also a mutant strain, CSI Delta, where the centromeres were unclustered. And what we observed is a cloud of IDU incorporation emanating from the SPB centromere, which is more dispersed in the mutant. We then performed another experiment where we tagged the SPB and a number of different origins in all three chromosomes of fission yeast. By measuring the three-dimensional distance between early and late firing origins in all three chromosomes of fission yeast, we identified that early firing origins were located closer to the SPB than the late firing ones. So overall, the results indicate that the SPB acts as a positive pole of replication initiation and that it drives origin firing by particle phosphorylation. Next, we assessed the model's sensitivity with respect to the different parameters, and we looked at DNA replication completion time that, as I mentioned before, we know from experiments to be approximately 20 minutes. What you see here is how completion time changes for different values of the number of firing factors, the number of activation factors. And you see that as the number of particles increases, then completion time decreases exponentially. We examined as well the effect of other parameters, and we saw that they're not so important, but the model is actually involved also with two different probabilities, an activation probability and a binding probability of the particles. So what we saw is that as we lower both activation and binding probability, the desired completion time of 20 minutes can be met simply by introducing more particles. So overall, our results indicate that the number of firing factors is a determinant factor of replication completion time, but this completion time is actually highly tunable for any value of n if we lower the probabilities. Last, we use the model to derive DNA replication kinetics, such as the one I'm showing you here, and we examined a broad spectrum of different combinations of probabilities. What we saw is that by plotting the number of firing factors over time, this number peaks in middle S phase when we assume approximately 300 particles and low values of our probabilities, which is an observation that fits experimental data that indicate a peak in middle S phase. Thanks a lot for the attention, and I hope you enjoyed the talk. I'd like to thank the people behind the in silico and the experimental work that I presented to you today, in particular Jonas, Amelia, Patrula, and Eve. If you're interested in the work, please check out our preprint as well as our GitHub repo that contains all the source code and the data. Thanks a lot.